Hello, everyone. Thank you for attending today's webinar, Supporting Clinical Trials with Early Detection Biomarker Testing, presented by Quanterix. I'm Rebecca. I'll be hosting today's webinar. I would like to introduce our speaker first. We'll be joined by Mingwei Zhao, Senior Director of Accelerator Services at Quanterix. You can find his full bio under the Speakers tab on the left side of your screen. We just have a few housekeeping notes before we get started here. You can access additional materials from today's presentation by clicking the Handouts tab on the left side of your screen, and you can access closed captions from the bottom right corner of the video player. The webinar is being recorded. It will be available to watch on demand within 24 hours. And during the presentation, you can submit any questions you have using the Q&A tab on the left side of your screen. We will try and get to those at the end. All right, let's go ahead and get started. Dr. Zhao, please go ahead.
Thank you, Rebecca. That's a very nice introduction. Hello, everyone. I'm Mingwei Zhao. It's really my great pleasure to be here today to join the webinar to share with you all about how Quanterix has been using ultrasensitive biomarker testing to advancing the early detections in clinical trials. So in my today's presentation, I would like to cover 4 areas. So the first is going to be a brief introduction about our Quanterix ultrasensitive technology. And the second part is really just introduce our offerings from Quanterix and also the accelerator services. And the third part is going to focus on the biomarker testing services we provided in Accelerator Lab, especially the premium service. And then the last section, I would like to just highlight a few examples about clinical trials and clinical studies we have been supporting using our ultrasensitive biomarker testing. So let's start. As most of you probably know, that Quanterix has been focusing on developing ultrasensitive protein detection technologies over the last 17 or 18 years. And then you might have questions that if the traditional ELISA is already good enough to be able to measure a lot of proteins in human biofluid, then why Quanterix and a few other companies still investing to develop an ultrasensitive platform? So the answer is, although there's a lot of proteins can be measured using traditional ELISA or other technologies, there are still thousands and thousands of proteins which are at very, very low concentration in the human samples. And then those proteins cannot be easily measured by traditional method. And a lot of them are tightly associated with diseases. That's why detecting them and measure them from the human biofluid is critical to be able to either use as a great biomarkers or to monitor the disease progressions or, identify more targets for drug discoveries. So the technology Quanterix has been developing is called Simoa technology, which stands for single molecular array technology. Due to the time today, I'm not going to go through the technical details how we developed this technology. But one key area I want to highlight is the key innovation of this technology is we developed microvalve chips. And then this array contains more than 20,000 microvalves, which can hold, each valve can only hold one single bead, and then about 50 femtoliter liquid. And by developing this microarray, we are able to isolate the beads into a single valve. And the background is super low for this system. That's why it enables the detection of the signal even from a single enzyme. That's why for Simoa technology, we can push in the sensitivity even further compared to traditional ELISA. And as you can see, for traditional ELISA, typically the sensitivity is around nanogram per mil to probably picogram per mil with ECL and a few other new technologies. However, for Simoa technology, we're really pushing the sensitivity to a femtogram, even attogram per mil sensitivity. And then with this great sensitivity, we are able to measure a lot of new biomarkers and protein isoforms. As you all know that there are about 20,000 active coding genes in human genome, and then those transcribe to more than 1 million protein isoforms. Historically, a lot of proteins isoforms, even post-translational modifications, of those proteins cannot be easily measured. But with the sensitivity Simoa technology provides, we're able to measure those proteins and really unravel the heterogeneity of the biological issues, and also identify new targets and measure them to help the diagnosis and also drug discoveries. And then leveraging the ultra sensitivity of our technology, we are able to detect the proteins in a non-invasive way compared to the traditional method or CT scans. So moving to the next slide, this slide is basically introduce the products and service we're providing to our customers. Over the last 18 years, Quanterix have developed not only instrument, but also assay kits. And now we also have services. So for the left column, you can see Quanterix have developed 3 instruments, HDX, SRX, and SPX. So the HDX and SRX are really focused on the beads-based technology, and the SPX is for the planar array technology. And within the 3 instrument, the HDX is a fully automated instrument which combine the ultra sensitivity technology and also liquid handling system. This not only provide the great sensitivity, but also provide the great precision as well in the test. And then the SRX and the SPX, those 2 instruments are semi-automated instrument, which can maintain the same sensitivity, but also provide a lot of flexibilities for users to develop their own assays. And for assay kits, over the years, we have developed a lot of assays on our menu, which covers neurology, oncology, infectious disease, and immunology, etc. And not only the ready-to-use assay, we also provide homebrew kits, which enables a customization of your own assays. And then the assays we developed includes not only single-plex assay, but also multiplex assay as well. And on the right side, you can see we also provide services. There are 2 parts of services we're providing right now. One is the contract research service through our Simoa Accelerator Laboratory. And then the second service we're providing is the routine diagnostics, which include the LDT assays of p-tau217, p-tau181, and NFL. And with all the products and service we're providing to our customers in the field, we're really covering from discovery to diagnosis. And then from the right side, you can see with the instrument and the assays we developed, we're really helping the customer research. We have right now more than 1,000 instrument placement in the whole field. And also, the technology has been used in more than 2,900 publications. And then on the right side for the diagnostics, we're really providing the RDT service in our CLIA lab, and then to help diagnostics. And then in the middle, the clinical trials is what I will probably be more focused on today. And then the clinical trial support is through our accelerator services. And then I will spend the next few minutes to introduce our accelerator lab. So what is Accelerator Lab of Quanterix? It's actually a pioneer partners for clinical research and drug development pipelines through ultra-sensitive biomarker testing. And then initially, the Accelerator Lab was established as a demo lab when we start commercialization of the technology. Because the instrument is relatively big, so our commercial team won't be able to carry it over to customer sites easily. So that's why we established Accelerator Lab to really provide the early and easy access to our customers so they can have a taste of the technology. And soon we realized that Accelerator Lab can do more than demo. Then we start building the team, getting a lot of experience, and right now we have more than 150 years of assay development experience, driving breakthroughs in biomarker discoveries and control studies. And then Accelerator has become the innovative solutions for all our customers. Over the years, we have completed more than 2,000 projects supporting the full spectrum of clinical research and drug discoveries. And we have more than 500 customers globally, including leading biopharmas and academic institutions. And right now, we're aiming to turn the accelerator into really the engine for accelerated development, which enables a faster, more sensitive diagnostic solutions to meet complex clinical needs. And then for accelerated service, it contains two major functions. One is the customer assay development and research, which we call care team. So within care team, where the main focus is to develop custom assays and to meet customer needs. And over the years, we have experience with hundreds of biomarkers in a wide variety of species and matrices. And we're very strong in neurology, but also we have expertise in inflammation, oncology, and infectious disease. We not only develop biomarker assays, but also PKPD, ADA, and competitive assays to support drug research and development. The second function of accelerator team is the biomarker testings. And then for the sample testing group, we take projects spanning early proof of concept through late-stage prospective clinical trials. And then we provide service and offer service of Quanterix full menu. And then we also have our dedicated QA team, and then we provide different levels of validations, even clearer levels and validations follow CLSI guidance. And then the accelerator lab right now has more than 50 staffs. And then the team is still growing. And then we have a team of project management, and we have senior staff experienced in assay development, validations, troubleshootings, and the regulatory applications. And then we have staff with LCP certificate for clinical testings. Right now, our facility is about 4,500 square feet, and we have 25 HDX instruments focused on the beat service, and 8 SPX instruments focused on the planar service. And then we are the biggest Simoa service providers in the whole world. And we have our LIMS system and also a CLIA certified laboratory. And we have our dedicated QA quality assurance team in Accelerator, and we are GCLP compliance. And we also have ISO 15189 accreditation. And we also have a clear laboratory directors on board. And then next few slides, I would like to highlight our biomarker sample testing capabilities. And then right now in our accelerator lab, we can take approximately 100,000 samples per year. And then we saw significant growth year to year, and because of the broader adoption. And also we have the capability to increase the testing to 3 times more. And then we have active project management to support testing needs as the trial goals evolve. And we provide full service menu to our customers. And then this slide is trying to show you the service tiers we have for our biomarker testing services. And then the left side, you can see that's the standard RUO service. And then that's designed for exploratory research, and there's economic options for customers to get access to Simoa platform. And also, although the data quality is good, however, we do not recommend to use the data for clinical trial enrollment or decision making or regulatory applications. Certainly, we can provide a customized RUO service for our customer, which include, for example, endogenous controls, lot-to-lot bridgings. However, that includes additional cost. And on the right side, as I mentioned earlier, we have LDT service really focused on diagnostics, and then will incorporation into medical records, and then for inclusion of clinical trials. But today, I would like to more focus on the tier in between, which is called premium service. So the premium service is designed really to support clinical research and therapeutics trials, and then which require the higher levels of data integrity. For premium service, we include endogenous controls for every test, and then we make sure the incoming kits has met the QC criteria, and we also have the bridging of the kits to make sure the performance of the kits is consistent over the years. And also we have access to our QA team, and then which can help interface directly with FDA if necessary. So I want to expand a little bit more about the premium service we have. Prior to launch premium service, we for most of the project we did is REO standard service, which we sometimes need to tailor specific to sponsors' individual needs. Although we can do that, there's additional cost associated, as I mentioned earlier. It's expensive and also it's time consuming because we need advanced planning. And also it's subject to errors because this is not our standard REO service and the workflow and the process is different. So, and based on the experience over the years, we noticed that there is more and more demand of, service higher than just REO standard. And a lot of customers, when they start their REO service in our accelerator lab, and then they chose the economical and simple path. However, when the studies move on, they realize that they need to harmonize the data, the future batch of the data with previous studies. Then there is always a gap and challenges there. And only we notice that only certain sponsors has the support staff which can make the right suggestions about what validations you need, what controls you need to use, all those things. So that's why, with all those gaps and challenges, we decide that maybe we should just upgrade our service into the next level. So that's why we launched the premium service. And the post-launch, a lot of our customers choose to upgrade their REO service to premium service because they really see the benefit. And with the standardized method and acceptance criteria established, that really allows the lab to efficiently process the samples and offer increased efficiency. And then I would like to explain why premium service can promote higher quality of data. As a lot of you probably know that there are always the source of biases in the biomarker testing. The sources coming from a lot of areas. For example, sample collection method, storage conditions, and laboratory conditions, operator to operator difference, instrument to instrument, and assay lot to lot difference. So all these single pieces of bias will add noise to the accumulated data. And then in the end, inhibiting really quantify a statistically significant change across the longitudinal studies. That's why we launched the premium service to really counter, control, and quantify those bias during our testing. How are we going to do it? First, we perform an analytical validation for following CLSI guidance, and then to really characterize and demonstrate the assay robustness, and then try to identify the potential source of interference. We also added the benchtop stabilities for sample to really monitor the sample stabilities during the handling. And we also provide the standard collection method. And in the lab, only ASCP certified scientists with hands-on training and experience can offer the premium service. And the instrument we use for premium service are only a clear certified instrument and maintained. And also for each of the testing runs, we include endogenous controls with predetermined acceptance range, which really monitor the plate-to-plate bias. And also all results were provided to the customers will have to ensure duplicates. That actually will help monitor the precision of the reported concentration. And all reagent we received in Accelerator Lab for premium service, there will be incoming material QC, and only the kits meet the acceptance criteria will be used for the testing. And all the different lots of the kits will be bridged to the anchor lots to maintain the consistency of our testing over the years. It's regardless of the sponsors or trials. And then, as I mentioned earlier, over the years, we have seen, keep increasing demand of the premium service. And what has been driven the immediate adoption and the increase of premium service? There are a few things. First is, of course, premium service offers tremendous values driven by the added rigor and quality. And second is now FDA recently afforded drug approvals based on biomarker data alone. And quite a few assays and markers have been determined as a must-have for given trials. For example, p-tau217 for Alzheimer's disease trials, and NfLs for neurodegenerative disease. And the successful trials will always submit to regulatory agencies, and our premium service will actually leverage our quality QA team and directly interact with the agencies to help the sponsors to be able to submit the data to FDA. And with all this being said, I mean, the premium service is really the next level of service we're providing to our customers. So the next few slides, I would like to just highlight a few of examples which we used over the years. We used our accelerator premium service to support clinical trial studies. The first one is the p-tau217. As you all know, that p-tau217 is one of the best tests, standalone tests for Alzheimer's disease testing. And not only Quanterix, a few other companies, they also have p-tau217 assays and tests. However, while most of the p-tau217 tests right now on the market can achieve very high accuracy and specificity and sensitivity, meaning the accuracy values, the best clinical utility is still when all the patient can receive the results. And based on our experience, we have used our Simoa p-tau217 assays in Biohermes trials and the VOMC trials, and we measured more than 2,000 samples. And our sample measurement rate is 100%, meaning we can provide results to all of our patients. However, a few competitive assays, for example, mass spec and chemiluminescence, although, again, they provide high accuracy, but based on the reports, there's typically about 30 to 40% of the patient which cannot receive the samples due to various different reasons, sensitivities and testing and all, etc. So that's why right now Simoa is still the most reliable technology on the market, which can provide the most meaningful and results to our patient. And then this slide shows the example of using NFL, which is neurofilament light, as the key biomarkers to support clinical trials and research. On the left side, you can see Annexon Biosciences, which is a pharma company. They chose to partner with Quanterix Accelerator Lab and testing NfLs for their pivotal phase 3 trials for their drug ANX005. And then those are the drug developed for the Guillain-Barré syndrome. And then the testing results is very promising. It shows early reduction in NfLs for nerve damage versus placebo controls between week 2 and week 4. And these results really supporting Annexon for their drug development. And on the right side, you can see most recent publications highlight the promising biomarkers NFL and GFAP in the screening of the brain tumors in patients with lung cancer. As many as 40 to 50% of lung cancer patients will develop the brain tumor towards the late stage. That's why using biomarker, blood-based biomarker, leveraging Simoa ultrasensitivity will have the potential to identify and monitor the disease progression and identify brain tumor at early stage. And then this is another example of a drug approved by FDA, which is supported by Simoa technology. And then the tofersen, which is a very promising drug for SOD1 associated ALS disease. And then back in 2021, when Biogen performed the phase 3 studies using clinical measure alone, did not yield a great benefit. And then that's why the trial actually failed. And then Biogen chose to partner with Quanterix and using NFL as the biomarker, and then to provide evidence to see the reduction of NFL in the plasma. And this actually greatly helped their their trial submission and lead to the FDA accelerated approval of this drug for LS treatment in April 2023. So this is a great example to show using ultrasensitive biomarker testing and blood-based biomarker, this our Simoa technology really can support the drug discoveries. And this the approval of this drug also gave a lot of ALS patients the hope that ALS is a treatable disease. So that's really encouraging. And then this slide shows the examples of another few FDA-approved drugs recently. Over the last 20 years, you probably know that there was only 2 drugs approved by FDA for Alzheimer's disease, the lecanemab and donanemab. So those 2 drugs are disease-modifying drugs, which are very promising to treat early-stage Alzheimer's disease. And the other couple drugs was approved as well within the last 3 years. So all the 4 drugs approved by FDA all fueled by Quanterix ultrasensitive detection technology. And then not only help them to approve the drug, but also it create a lot of business opportunities for Quanterix as well. And looking at 2024 and beyond, right now there are more than 120 biopharma CNS clinical stage programs are active, in which 24 programs are specifically focused on Alzheimer's disease. So there is a lot of opportunities for our company and the ultrasensitive testing service in supporting the clinical trials and studies. And then this slide is really to highlight the examples of clinical trials we have supported over the years. As I mentioned, Lilly donanemab, and then the Biogen Eisai lecanemab. Those are the 2 great Alzheimer's disease drug. Then they all use the Simoa p-tau217 or p-tau181, GFAP, and NFL as the biomarkers to support their trials. And then the p-tau217 is also used for Janssen drug trials as well. And then for Novartis, Quanterix offered the VGF assays in Accelerator Lab to help their ranibizumab, the monoclonal antibody drug, which to support their macular degeneration disease. And then for Alnylam, right now the phase 3 trials for the patisiran drug, which is ongoing, and then the results is really promising. So with all those highlighted trials and examples, probably you will get the sense that Simoa technology is really a great tool to support the clinical trials. And then towards the end of my slides, I would like to also highlight the utility of using Simoa technology for patient selections and also the target enrollment for clinical trials. As you can see on the left side, for representative clinical trial design, for example, if we want to design a trials for looking at the Alzheimer's disease drug, and then maybe the inclusion criteria would be cognitive unimpaired individuals, which were identified by PET scan. And then the target enrollment number is probably around a few thousand subjects. However, the positive scan rate is only 1%. And then that means in order to get 3,000 subjects for the enrollment, probably the sponsor needs to screen probably close to 300,000 potential subjects to be able to identify those 3,000 subjects. As we all know that PET scan is expensive. It's also invasive and not very accessible. And then to do one PET scan, the cost is around $5,000. And then that's create a huge burden for the enrollment cost, which close to $1.5 billion for just the enrollment screening. However, if we use Simoa technology, the cost will be decreased significantly, about 50-fold. And then that not only can provide the advantage of the enrollment cost, but also the test is simple and also is non-invasive and quick. So that's why for Simoa blood-based testing can enable the non-invasive, cost-effective identification of patients really suitable for the clinical trials and accelerating the trial enrollment and increase the possibility of approval. So the last couple slides, I would like to highlight what we have done recently to really further improve our premium service in our accelerator lab. So p-tau 217 is a great example. As I mentioned earlier, a lot of companies right now, they do provide p-tau 217 test. However, by using p-tau 217 test, because the concentration is so low in the the patient sample, there was two cutoff strategies being used, meaning below the low cutoff, the patient has been identified as a very low risk of Alzheimer's disease, but above the high cutoff, the patient has a higher chance of getting Alzheimer's disease. However, there is an intermediate zone, which is about 30% of the results or individual tested, which they have the values fall into the intermediate zone, which they need further tests to be able to identify the risk. So for Quanterix, we launched recently the multi-marker test, which include p-tau 217, amyloid beta 4042, and also NFL and GFAP. With the multi-marker algorithm, we are significantly reduce the intermediate zone for the Alzheimer's disease testing, which means we can cut down the 30% intermediate zone to close to 10%, which means more and more patients will get a more clear testing results for their either diagnosis or treatment. So that's the significant advantage of using our multi-marker algorithm testing, and it's going to be launched in our accelerators premium service very soon. And then for future capabilities, p-tau 217 is definitely one, multi-markers. We also recently launched the BD-tau, which is a brain-derived tau for RUO service in Accelerator Lab. And we're going to provide the BD-tau premium service in first half of 2025. And then besides BD-tau and P17, now we also provide the new p-tau 231 plasma assays. And then this assay will be launched very soon in Accelerator as well. And including the BD-tau, we also launched a multiplex assay, which we call N4PD Advantage Plus, which includes NFL, GFAP, BD-tau, and UCHL1. And then for other multiplex, we recently launched 3 4-plex assays, which will achieve great sensitivities for cytokine testing. That's our, the assays we're going to launch in premium service for cytokine measurement to monitor oncology and infectious disease or immunology very soon. So with all these capacities, we're seeing a great and bright future for our premium service in Accelerator Lab and in general, our biomarker testings to support the clinical studies and trials. So I would like to end my presentation by emphasizing that Simoa technology not only providing the ultra sensitivity, great precision, but with a premium service we're providing, we're actually really giving our customers the high level of high level data with great integrity, and then leveraging Simoa technology. Our aim is to catching the disease early and then in a non-invasive way, so which will enable the healthcare instead of today's sick care. If we can detect the disease earlier, treat the disease earlier, and even prevent the disease earlier, and then the outcome will be much better than just developing drugs and treat the sick patient as what we're doing right now. So that's why I would like to end the presentation, and we're looking forward to working with everyone in the future. And then I would like to thank everyone for your attention, and we have the contact information at the bottom of the slides. And then feel free to contact us if you have any questions or inquiries about the technology, accelerator service, premium service, or in general. And then now I'm going to open the floor for questions, and I'm happy to answer any questions. Thank you very much.
All right. Well, thank you so much. We are ready to move into the Q&A section right now. I see we have a lot of great questions already. We'll try and get to as many of them as possible in here. To begin, how long does your contracting process take in order to initiate a new project?
That's a good question. So typically, I mean, the process is the customer will contact us and through our regional sales managers, and then we will actually set up a meeting with our project management team, and then we're going to discuss really to understand the goal of the studies, which tier of service we're going to use, and normally we'll try to set up the meeting as quick as possible. And once we understand the needs and the scope of the project, we normally will either provide a quote in 1 to 2 days if it's a small project. If it's a larger size project or longitudinal studies, and then we would like to provide a written SOW, which is statement of work, and then that typically takes about 1 to 2 weeks. Once everything is done, and then when we receive the PO, and then we receive the sample, we can start the sample testing service as quick as possible.
All right, thank you. The next question is pretty technical here. Forgive me if I fumble this a little. Shouldn't tau also be on the multi-marker assay for AD ratio tau over p-tau 217?
That's a great question. Right now, our multi-marker assays is really leveraging our p-tau 217 and also N4PE, which is the GFAP, NfL, and A beta. And then we feel like the p-tau 217 is one of the best standalone assays for Alzheimer's disease testing, and combining with NfL and amyloid ratio, I think that provides a great values to look at the risk of Alzheimer's disease. The tau and the total tau, I know for mass spec, people look at the p-tau 217 versus tau ratio. That's also have great values, but right now for our multi-markers, we feel like the NfL, GFAP, and A beta combining with p-tau 217 is a great combination, which really, as I mentioned, cut down the intermediate zone from 30% to 10%.
All right, great. And just a reminder to our audience, you can still submit questions. We are happy to take those. Moving on, if we decided to choose Quanterix, how quickly could you provide us single biomarker data for 5,000 samples?
That's a good question. We love the business of 5,000 samples. And then normally for the scale like 5,000 samples, it takes about probably 10 to 12 weeks. But it depends on a lot of factors. For example, how soon we can get the contract initiated, how soon we receive the PO, and whether the samples are ready to be sent in batch, and whether we need to spend more time to sort the samples, and also wait for different visit and wait for to batch samples in the same plate. All those variables will affect the timeline. So that's why, although typically it's about 10 to 12 weeks, but it really depends on a specific project. And we would highly encourage customer to reach out to our project management team, so we can really better understand how soon we can receive the sample, how soon we can start the project, and then we can provide a more accurate timelines for the project.
All right, next question. How much volume do you typically need to perform biomarker testing services?
That's a great question. I would say a lot of our customers, they start with the same question. Sample volume, we understand sample volume is always a challenge for a lot of studies. Typically, for most of our small assays, we require samples less than 100 microliter for our typical assays and dilution fold. I would say if you target 100 microliter, that would be sufficient for a single test with duplicates. However, we highly recommend customer to consider providing us 2x volume, just in case, if anything, we need to repeat. Although the repeat rate is still low, based on our experience, the repeat rate is less than 5%. However, if anything happens, we would like to have the sample ready to be able to directly repeat the test. so we don't have to run into the delays, additional shipment, sample volume, and adequate, and all those challenges. So that's why, again, if customer can discuss all those details with our project management team, and then really figure out what will be the most meaningful volume to provide to the team, I think that would be very helpful.
All right. When will the multi-marker assay for AD be available for use in clinical studies?
I think it's going to be Q4 this year. I think that's the plan. Yeah, which is this
Next
quarter.
question.
Yeah.
Okay, wonderful. Does the Simoa assay require a serum or plasma sample?
We have different assays developed for different disease and research area. For neuronal assays right now, majority of our assays are really focusing on plasma sample. And then that's really to establish a blood-based biomarker detection to create a non-invasive detection method. And then for a lot of cytokine assays we have, they will be validated in serum plasma. And for certain assays, we of neuronal assays, we also provide serum testing as well.
Next question. What's the recommendation for EQCs in trials? Quantity, quality, source of matrix, etc.
Yeah. So I would say the EQC is a very critical piece for to maintain the integrity of the data and monitor the plate-to-plate variations and bias. So EQC, of course, stands for endogenous control, right? And then typically what we are going to do is we will talk to the customer, understand the goal and expectation of the scope, and we typically include two EQCs in the runs in the premium service. And then most of the cases, the EQCs will target the decision-making concentrations. We don't want to just randomly set up two concentrations which far away from the majority of the testing results. And then how we're going to choose the EQC, it depends on the matrices, availabilities, disease status, concentration of the analytes and the markers. We'll try to establish the most meaningful EQCs to monitor the testing. In terms of the quantity, as I said, most likely, most of the case we provide two EQCs, but sometimes three EQC. Really depends on the discussions with the customer. And also matrices, we would like to use endogenous samples not spiked with anything. That will be the best way to monitor the testing and be most relevant matrices to customer samples. And also the EQCs will have the predetermined concentration range. And then we typically run 10 runs to establish the range for EQCs before we start the customer sample testing. And we use that concentration range to really monitor the EQC performance on each plane.
All right. There's a follow-up for an earlier question about using serum versus plasma samples, and they ask about LiHep plasma or lithium heparin plasma.
Yeah, I think that's a good question. A lot of our customers actually ask about lithium heparin plasma. Right now, majority of our assays are validated in EDTA plasma. And then for heparin plasma, if you have samples already collected, we would recommend to do either a fit for purpose validation to really verify the assay performance in the matrix before we go too far to measure your clinical samples. And minimally, we should look at the matrix effect, specificity, dilution linearity, those aspects to make sure the assay really function as expected in heparin plasma, because it's not a validated matrix for our manual assay. But we certainly can do that, and that's why in my previous slides I was I was also highlighting that we provide all different kinds of validations, follow different guidance to really make sure we provide the overall solutions for our customers.
Speak about the workflow
Hello?
from sample in a tube to plate in instrument.
Sorry, you were cut off.
Oh,
Can
I'm
you repeat
sorry.
the question?
I have a mute problem. The question was, can you speak about the workflow from sample in tube to plate in instrument?
Sure, yeah. And then typically, as I mentioned, we start the collaboration with customers by the pre-sales meetings. And then we would like to understand really the goal of the project and expectations and sample type and how many samples. And then we'll provide the right quote, SOWs, to make sure we have a mutual understanding about the project. That's the first step. Once we have the project initiated, then we will talk to the customer, make sure we have the sample received in the right conditions and as expected, and also the timeline for different batch of samples from different sites. And then once we receive the samples, our lab assistance team will review the samples and monitor the sample receiving conditions, log into our LIMS system, and corresponding to the manifest we received to make sure all the samples, barcoding, sample type, and sample volume, sample numbers are accurate, matching the manifest. And then once the sample are received, and then internally, our project management team will work with the lab team, and then to schedule the testing runs based on the resource and the capability and the availability of the instrument. And then, I mean, before we start the testing, all the documentation, including working instructions, testing protocols, training personnel, we want to make sure everything is in place. And then when we start the testing, we'll take out the samples, we'll log out the LIMS system from the LIMS system, and then we'll samples in the right conditions, follow the protocols, and then we will dilute the samples, record all the details in the work instructions, and place them in the instrument, and then for the testing. And when the sample handling is done, samples will be put back into our freezers and logged into the LIMS system to record all the details. And then sample sawing, handling, and dilution schemes and calculations, all the details will be recorded in the work instructions and subject to audit. And then once we get the results from the instrument, we will export the results using our qualified software and doing the data analysis. And then we will the senior level scientists will review the results, and then to determine the validity of the results and the testings, look at the EQCs, controls, and calibration curves, make sure everything meets the criteria, and then we can release the results to the lab directors. Once they review and sign off, the data will be shared with our customers. I hope that answered the questions about the workflow and process.
All right, yes, that was very helpful. I think we are coming to the end of our time here today. If you have additional questions, you can contact our team using the emails that you see on the slide in front of you. And thank you all for joining us. Thank you to our speakers today. And thank you for joining this Fierce Biotech webinar. This webinar has been recorded. You will be able to access the recording within 24 hours by using the same audience link as today. Thank you again for attending, and we look forward to seeing you at future events.